Hello everyone, myself Kirti Gaur from the Department of the Paramedical Sciences of Rama University. My designation is tutor and I am going to explain you the topic is sterilization and disinfection. First, I am telling you the introduction part. Microorganisms are responsible for the contamination and infection. So that we have to kill them or to destroy them, there is the technique which we use is sterilization and disinfection. And you know that the bacteria are all present all around us, so that they cannot infect us. So we have to kill them. So there is a many process I am going to explain you today. So first is sterilization, disinfection and uses types. There are the many types of sterilization. Sterilization is of two types, which one is physical method and second one is chemical method. Today I am going to explain you only the physical method. In physical method, there is a sunlight, heat, filtration, radiation, ozone. These all are methods used in the physical method. And in heat is all further divided into two: dry heat and moist heat. Filtration, in this filtration there is two types of filtration are used. First one is depth filter and second one is membrane filter. Now in radiation, I will going to explain you in radiation that ionizing radiations, non-ionizing radiations, and last one is ozone. Now comes to the sterilization. What is sterilization? Sterilization is the process in which we have to kill the bacteria, viruses and viable spores, so that they can disinfect the surface area and environment and human body also. And the instrument disinfection is to destruction of the microorganism which they are present on the surface of the body, or it is present on which surface of body and the mediums. Now comes to the next uses. There is a many types of they use in many fields. First one is surgical fields. It is sterilization used in the surgical instrument to kill all the bacteria, so that the surgical instrument which we used in the medical field. And next one we use in the sterilization, we use for medium, culture medium, and for the reagents. And also we use in the sterilization, we use in the drugs and food, so that we can kill all the bacteria, so that we can secure them from the bacteria. Now comes to the types. I can explain you the types of physical method first. First is sunlight. What is sunlight? Why sunlight we use in sterilization? Sunlight is the most commonly and the natural process which we use for the sterilization. The all environment in the moisture is present. The moisture containing microorganism, in this the sunlight is very useful for this. In this, bacteria are killed by the sunlight rays, so that the environment is make the environment is bacteria free. And the next topic is heat. In heat, there is two types of heat. First one is dry heat and moist heat. In dry heat, there are many processes which we use. First one is red heat, second one is flaming, third one is incineration, fourth one is hot air oven. In red heat, in the laboratory we use the wire loop, needles and many other things which we use for red heat. Is to the things they are used, they are get held on the Bunsen burner of the flame, to get allow them to red heat. So that in red heat and flaming, there we use forceps and other things. We use glasswares and media culture media tubes which we allow them to Bunsen flame on them, allow them to Bunsen burner, not to get a red heat. So because to sterilize them. Incineration, in incineration, we infected materials are used to become destroyed by the heating, and kill them by the heat. And in incineration, hot air oven, in hot air oven it is used in a microbiological laboratory. It is widely used in microbiological laboratory, so that the bacteria are sterilized by the hot air oven, and the temperature is used in the hot air oven is 160 degrees Celsius for 2 hours. Now comes to the heat. Now comes to the moist heat. Moist heat. In moist heat, there are the various temperatures are used. First one is temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, and temperature above 100 degrees Celsius. Now in temperature below 100 degrees Celsius, there are various processes are used. First one is pasteurization, water bath. Next one is inspeciation. In pasteurization, there is a heat we use the mild heat to save the beverages components and the dairy products, so that they are free from the bacteria as they are in the pasteurization. Now comes to the water bath. In water bath, it is used for disinfection of a serum, body fluid, and vaccines. Now in inspeciation, heat on articles on which 3 successive days at 80 to 85 degrees Celsius for 30 minutes we use in inspeciation. Now comes to the temperature at 100 degrees Celsius. There is also a various process are used, just like boiling, steaming, sterilization. Now in boiling, it is very common in house also. Boiling is the process in which we use the atoms so that we kill the bacteria, as it is normally kill the bacteria but not the viruses. And in this we use for the boiling process is used for the 15 minutes. It is possible. It is useful for the media which are decomposed at a high temperature. So in autoclave, that's why it is used in a steaming process. Steaming is very useful only for media. Those who decompose in autoclave. And next one is sterilization. Sterilization it involves steaming at 100 degrees Celsius for 20 minutes for 3 consecutive days. Now comes to the third one. Temperature above 100 degrees Celsius. In temperature above 100 degrees Celsius, we use autoclave. Autoclave is a type of instrument which we used in the laboratory, which the temperature and timing is 121 degrees Celsius for 30 minutes. And in this autoclave, we use surgical instruments to sterilize culture media, autoclave, plastic containers, and plastic tube pipettes and other things which we use for this. And we also sterilize the glasswares also. Comes to the radiation part. Radiation is also of two types: ionizing radiation and non-ionizing radiations. Ionizing radiation contains gamma rays, X-rays, cosmic rays. They are very useful for the bacterial for the denaturation of DNA of the bacteria, so that it denatures the DNA and when it raptures the DNA of the bacteria, so they kill them with the rays, and this is very useful for the sterilization, and these all rays are very useful for in laboratories. Now comes to the non-ionizing radiation. In non-ionizing radiation, there is UV rays and infrared rays. They both are used in the surface to sterilize the surfaces of just like operation theaters and water treatment plant also, and other things also. So we all know about the sterilization of the physical method only. So and I can explain the chemical method in next lecture. So first, now recall all the things introduction. We have completed sterilization, disinfection, uses, types just like sunlight, I explained heat, dry heat, moist heat. In dry heat, I can read red heat, flaming, incineration, hot air oven. In moist heat, temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, and temperature above 100 degrees Celsius. Now filtration. I have not explained filtration. I leave that part. So I am going to explain to you. Filtration. The in-depth filtration is the most commonly used for the micrograde population in a solution for to allow them to pass through the filters. So it retain all the bacterial in the filter, and it makes the solution free from the bacterials. And in depth filter and the membrane filter, same in depth filter also and the membrane filter. In depth filter, the bacteria are retained on the depth surface of the bacteria, depth surface of the filter, and in membrane filter, the bacteria are filtered on the surface of the membrane, and this membrane is made up of synthetic materials, which is most commonly used in the laboratory. Now all topics is completed. Thank you.